All right, let's get started with today's deep dive and
Dive right in.
yeah, into the β-catenin pathway.
It's a good one. It's really central to so much of what we see both in normal development and in disease.
Yeah, I'm looking at all the sources you sent over,
Yeah.
and it seems like we're really going to try to understand
Yeah.
how this pathway works, like step by step.
That's the goal. And I think the first thing to really wrap our heads around is that this is a signaling pathway. So it's all about sending signals within cells.
Okay.
And the signals are sent using things called Wnt ligands.
Wnt ligands.
Wnt ligands, yeah. So you can kind of imagine these as like molecular keys
Okay.
that fit into very specific locks on the surface of cells.
Yeah.
And those locks are actually receptors, and there's two of them.
Okay.
They're called LRP and Frizzled.
LRP and Frizzled.
Yeah.
They sound like they're from Star Wars.
They do, a
Yeah.
little bit, but they're very important.
Okay.
These two receptors function as co-receptors, meaning they need to work together. In order to receive the Wnt signal, it's like a two-part code
Gotcha.
that needs to be cracked for the message to get through.
So when a Wnt ligand binds to both LRP and Frizzled at the same time,
Yes.
that's when things start
That's when the magic happens, yeah.
happening inside the cell.
It triggers a cascade of events
Okay.
inside the cell, and this cascade ultimately affects a protein
Okay.
called β-catenin.
β-catenin. I feel like that's the name that I see
It's a big one.
over and over again.
Yeah.
What's so special about β-catenin?
It's a very important molecule in this pathway. So β-catenin is kind of like a cellular switch.
Right.
When Wnt signals are absent, β-catenin is continuously being broken down. It's
Okay.
kept in check, right?
Right.
But when a Wnt ligand binds to those receptors,
Yeah.
it sets off a chain reaction that protects β-catenin from destruction.
So β-catenin is like running away from being destroyed all the
So
time.
it's like a game of tag, and the Wnt signal is like, okay, you're safe now.
Gotcha.
And so when Wnt signaling is active, β-catenin is protected
Right.
and starts to accumulate.
Okay, so it builds up.
It builds up, yeah.
What happens when it builds up?
So this is where it gets really interesting because β-catenin, when it's not being degraded,
Right.
can travel to the cell's nucleus,
Oh, into the nucleus.
which is the control center of the cell where the DNA is stored.
Oh, wow.
Yeah.
So this little protein just goes right into the nucleus.
It does. And once inside the nucleus,
Okay.
β-catenin teams up with other proteins
Okay.
to turn on specific genes.
Oh, interesting.
And these genes are often genes that control cell growth
I see.
and proliferation, basically telling cells when to divide and make more of themselves.
Oh, so I'm starting to see the connection to cancer now.
Yeah,
Because if it's always on,
right.
then you're going to have uncontrolled growth.
Exactly. And that's one of the main reasons why this pathway is a hot topic in cancer research, because mutations that keep the pathway stuck in the on position
Right.
can really fuel tumor development.
One of the sources specifically mentioned colon cancer.
Yes.
Is that a common one that's linked to this pathway?
Yeah, so a significant percentage of colon cancers
Yeah.
have mutations in a gene called APC.
Okay.
And APC is part of that machinery
Okay.
that's responsible for breaking down catenin.
Gotcha.
So when APC is mutated, it can't do its job.
Right.
Catenin runs wild.
So then it's just like always on.
It's always on. Yeah.
That makes sense.
Yeah.
So if we could find a way to control
Yeah.
this WAMP pathway, then maybe we could treat these cancers.
That's the hope, yeah.
Yeah.
Researchers are actively looking for ways to interfere with different parts of the pathway.
Okay.
Maybe block those ligands, maybe mess with the receptors,
Yeah.
maybe stop catenin from getting into the nucleus.
Wow, it's amazing to think about
Yeah.
how understanding these tiny little interactions
Yeah.
can lead to such big breakthroughs.
It's really quite remarkable.
Before we move on, I just want to reiterate for the listener,
Yes.
all these different steps,
Yes.
the Wnt ligands,
Uh-huh.
LRP, Frizzled receptors,
Yes.
β-catenin. It's accumulating.
Uh-huh.
It's going into the nucleus.
Yes.
It's intricate,
It is.
but understanding how it fits together is really
I agree, yeah. It's a beautifully complex system.
key.
But let's not forget, while
Mhm.
we're focusing on its role in cancer,
Yeah,
the β-catenin pathway is also essential for normal development.
right. So it's not bad. It's just,
powerful.
it has to be
Yeah, you need to keep it in check.
regulated.
Exactly.
Speaking of fine-tuning and regulation, one of the sources that you sent over talked about something called cyclic AMP signaling microdomains.
Right.
Are these like little neighborhoods
They are. It's a great way to think about it.
within a cell?
Yeah. So think of a city
Okay.
with different districts that have their own specialized functions.
Sick.
Microdomains are like those districts, but inside a cell.
Wow.
And they allow multiple signaling pathways to operate simultaneously
Oh, so
without interfering with each other.
cross paths.
Right.
Oh.
So you can have multiple things happening at
Well,
the same time.
How do they stay separate?
That is a great question, and that's where these special proteins
Okay.
called kinase anchoring proteins, or AKPs, come in.
Okay.
And AKKPs act like zip codes.
Wow.
They direct specific signaling molecules to their designated microdomain.
So they're like the traffic cops
They are, yeah.
of the cell.
They make sure the right signals get to the right places at the right time.
So organized.
It is,
Oh.
and this level of organization just underscores how dynamic cells are, how
Yeah.
responsive they are.
Uh-huh.
They're constantly adapting and responding to cues.
This reminds me of something we saw earlier
Yeah.
with the glycine receptors
Yeah.
in the spinal cord.
Yeah, that's a great example
It's like another example
of how important
of how precise signaling is
and
important.
inhibitory signals are, yeah.
Inhibitory signals.
So glycine is the main inhibitory neurotransmitter.
Inhibitory meaning it stops
It stops things.
rather than activates.
Exactly. So imagine your alpha motor neurons,
Okay.
which control your muscles.
Right.
If they were constantly firing,
Yeah.
you would have uncontrollable muscle contraction.
You feel like
You'd be seizing up.
yeah, you'd be very tense.
Right.
So glycine acts as a brake,
Okay.
ensuring smooth, coordinated movements.
So we need that.
We do.
Glycine.
Yeah.
The source also mentioned a closed, desensitized state of the glycine receptor.
Right.
What is that?
Think of it like a doorway
Okay.
that can be open,
Oh my God.
closed, or jammed shut.
Okay.
So the receptor can be closed and ready to respond to glycine,
Okay.
or it can be temporarily jammed, even if glycine is present. And this desensitization,
Which helps regulate how the spinal cord reacts to glycine,
Very fun.
preventing over-inhibition.
oh, so it's like another layer,
It is, yeah. It's like a safety mechanism.
the control,
Yeah.
so it doesn't go haywire.
Exactly.
Interesting.
And this theme of intricate regulation and balance
Right.
is something we see again and again
Yeah.
in cellular processes.
Right.
Including, as we'll explore further, the β-catenin pathway.
These examples, the glycine,
Yeah.
and then we were looking at
Yeah.
thyroid hormone.
Right.
They really drive home
They do.
how intricate this all is.
It's amazing.
All the
Beth.
cellular signaling
That tiny molecule
have a
massive impact,
Tem-
to huge impact
on what our cells do.
on what our cells do.
And this is also where things can go wrong.
Right. Yeah.
Mutations in receptors or
Yes.
signaling pathways
Uh-huh.
can disrupt normal development,
Right.
lead to diseases like cancer.
It brings us back to β-catenin.
It does, yeah.
Yeah.
Speaking of disruptions,
Yes.
one of the sources talked about this research with a modified form of a core pressor
Yes.
called Corey.
mCory. Yeah.
What is a core pressor?
So core pressors help silence genes.
Okay.
They essentially put a do not disturb sign
Guts.
on certain genes. But this modified form, mCory,
Okay.
disrupts that normal silencing process.
It flips the sign.
It does, yeah. It's
Yeah.
like throwing a wrench in the machinery.
So it's preventing genes
Yeah.
from being turned off
When they should be.
when they should be.
Exactly.
And so to study the effects of
Yeah.
this disruption,
Yeah,
they created transgenic mice.
they did, yeah.
Transgenic mice.
Meaning they've been genetically modified to have this extra mCory,
Gotcha.
and they overexpressed it in their livers.
Okay.
And this led to some interesting changes,
Okay.
including increased cell proliferation.
So more cell
Oh,
growth.
cell growth in the liver, yeah.
Wow. So it really throws things
It does.
out of whack
Yeah.
if you mess with that gene silencing.
It highlights the importance of these core pressors.
How did they figure out what genes
Yeah, that's a great question.
were affected?
So they used a technique called cDNA microarray analysis.
Okay.
And this is like taking a massive snapshot
Okay.
of the activity of thousands of genes
Wow.
simultaneously.
So they can see everything at once.
Yeah, it's like looking at the whole picture
Oh, cool.
instead of just one gene at a time.
That's really cool.
Yeah, it's a powerful tool for understanding
Yeah.
how changes in one part of the cell can ripple out and affect
Right.
many different genes and processes.
It's amazing what they can do
It is, yeah.
with all these microscopic things.
It's remarkable the level of detail that we can get
It is these days.
with
yeah.
the technology we have.
I feel like we've covered so much ground already.
We have, yeah.
But I want to make sure that we loop back to the β-catenin pathway itself.
Yeah.
We've talked about its role in
Mhm.
development
Right.
and disease,
Uh-huh.
but I'm curious,
Yeah.
how do researchers
Yeah.
actually study
That's a great question.
this pathway?
So there are numerous ways that
Okay.
scientists explore the complexities of the β-catenin pathway.
Okay.
One common method
Yeah.
involves introducing mutations into genes
Okay.
involved in the pathway,
Mm-hmm.
and they do this either in cell cultures
Right.
or in animal models.
Okay.
So they're basically creating
Okay.
controlled experiments
Got you.
where they can tweak specific components.
So we'll change a little thing.
You'll see what happens.
Yeah, and see what the consequences are.
Okay.
So they might expose cells to different Wnt ligands
Okay.
or block specific proteins
Huh.
in the pathway.
Gotcha.
This allows them to isolate specific variables
Okay.
and observe the effects on things like
Oh.
β-catenin levels.
Or gene expression. So it's a powerful tool for
Yeah.
dissecting the steps in the pathway.
So they start with that, usually.
Often, yeah. Cell
Oh.
culture experiments are relatively quick
Okay.
and cost-effective,
Got
so they're often a good starting point.
Okay.
And then you mentioned animal models.
Right.
Are those mainly mice?
Yeah, are they mice?
Yeah, so mice are commonly used
Okay.
because we can genetically modify them to
Right.
have specific alterations
Yeah.
in genes
Right.
involved in the β-catenin pathway.
So they might make mice
Yeah,
that either overactivate it
exactly.
or block it completely.
Yeah, and then they can observe how these genetic changes
Okay.
affect development,
Uh-huh.
tissue growth,
Right.
even disease progression.
Okay.
So they're looking at the consequences in a living organism.
That's wild.
Yeah, it's really quite powerful.
It's like you're actually seeing it play out.
Yeah.
That's so cool.
Like, for example,
Yeah.
researchers might observe that mice with overactive Wnt signaling
Right.
are more likely to develop tumors.
Yeah, exactly.
So it provides really strong evidence
Yeah.
for the role of this pathway in cancer.
It's incredible that we can study these things
It is.
in living creatures.
It really highlights the power
Yeah.
of these model systems.
While these cell culture and animal models are really valuable,
Uh-huh.
is it possible
Yeah.
to study the pathway in humans?
It's a good question. It can be more challenging,
Okay.
but researchers are finding ways to do it.
Okay.
One approach is to analyze biopsies
Okay.
from patients with diseases like cancer.
So they're taking a little piece of the tumor and
Sure.
looking at the pathway.
Yeah, they're looking for clues
Okay.
about how β-catenin is
Yeah.
behaving in those tissues.
So then they can be like, oh, okay.
Yeah,
It looks like it's really active here.
exactly.
Maybe that's why this is happening.
Yeah, and that information can then be used to
Yeah.
potentially
Okay.
help diagnose diseases
Hmm.
or even guide treatment decisions.
So it could personalize medicine.
Exactly, that's the idea.
Wow.
Yeah, personalized medicine
That's amazing.
where treatments are tailored
Yeah.
to an individual's unique molecular profile.
Wow. It's amazing to think that
Makes sense.
understanding these tiny pathways
Yeah,
could lead to these big advancements.
it's pretty remarkable. And as our knowledge of the β-catenin pathway grows, we can expect to see
Yeah.
even more applications in personalized medicine.
It seems like we've covered so much.
We have.
This has been a really enlightening discussion so far.
I agree.
We went from the very basic steps
Yeah.
to its roles in development and
Right.
disease,
Right.
and now we're even talking about
Yeah.
using it as a therapeutic target.
And there's still so much more to explore. Oh, I can't wait.
Yeah.
I'm so excited for part 2.
But for now,
Yeah.
we'll have to take a short break.
Yes.
Stay tuned.
Stay tuned.
Welcome back to our exploration of the β-catenin pathway.
It's good to be back. I'm really enjoying this deep dive.
It's a fascinating pathway, that's for sure.
I was thinking during the break about how we can actually study this
Right.
in a lab setting.
Yeah. So, I mean, it seems so intricate
Right.
and microscopic. How do we...
It's hard to even picture.
It is, but scientists have developed some really clever ways
Okay.
to study this pathway.
Okay.
And two of the most common approaches are cell culture experiments
Okay.
and animal models.
So cell culture, that's like growing
Exactly.
cells in a dish.
Yeah, so it's like creating a mini laboratory
Okay.
where researchers can control the environment
Right.
and manipulate the cells.
So they're kind of like
Yeah.
putting them in different situations,
True.
seeing how they respond. Yeah, so they might expose the cells
Okay.
to different Wnt ligands,
Okay.
or they might try to block
Right.
specific proteins in the pathway.
So they're manipulating things.
They're manipulating things
They're
and
kind of-
seeing what happens.
Gotcha.
And this allows them to isolate specific variables
Okay.
and see how those variables affect things like
yeah.
β-catenin levels or gene expression.
A very controlled way.
It is a very controlled way to
Yeah.
study the pathway.
That makes sense.
And, cell culture experiments are relatively quick and cost-effective.
Makes sense.
So they're often a good starting point for WNT research.
And then you mentioned animal models.
Yes.
So that's usually mice.
Usually mice, yeah.
Mice, okay.
Yeah, so mice are commonly used because we can genetically modify them
Okay.
to have specific changes in genes that are involved in the β-catenin pathway.
So they might make a mouse that like overexpresses or a mouse that's blocked.
They might create a mouse that has a mutation that activates the pathway all the time.
Right.
Or they might make a mouse that has a mutation that blocks the pathway completely.
And then they'll watch what happens.
Exactly.
Gotcha.
So they observe how these genetic changes affect the mouse.
Yeah.
Things like development, tissue growth.
So they're really seeing it play out.
They are.
In a living organism.
They're seeing the consequences of these manipulations.
That's amazing.
It's a very powerful approach.
So cool.
For example, researchers might find that mice with overactive WNT signaling are more likely to develop tumors.
That makes sense.
Right. So it provides strong evidence for the role of WNT in cancer.
It's really cool that we can
Yeah,
study these things in living creatures.
It is, yeah.
It really is.
It highlights the power of these model systems.
So we have the cell cultures.
Yes.
We have the animal models.
Yeah.
Is it possible
That's a great question.
to study the pathway in humans?
So it can be more challenging.
Right. Because you can't just manipulate things.
Exactly. You can't manipulate things ethically in humans the same way you can in mice or in cells.
But there must be ways.
There are ways, yeah.
Okay.
One common approach is to analyze biopsies from patients with diseases like cancer.
So you take a piece of the tumor.
Exactly.
And you look at the pathway.
You look at the pathway, yeah.
Okay.
And you're looking for clues
Okay.
about how β-catenin
Right. Is it active?
is it active?
Is it not active?
Is it not active?
Is that why this is happening?
Yeah, and that information can then be used to potentially help diagnose the disease or even guide treatment decisions.
That's where you get into personalized medicine.
Exactly. That's the hope.
Yeah.
Personalized medicine.
Wow. It's amazing to think
It is a very exciting area.
that understanding these tiny pathways could lead to such big advancements in healthcare.
Absolutely. And as her knowledge of this pathway grows, we're going to see even more applications in personalized medicine.
It seems like we've covered so much.
We have.
This has been a really enlightening discussion so far.
I agree.
We went from the very basic steps of the pathway to its roles in development and disease.
Yeah.
And now we're even talking about it as a therapeutic target.
It's a very exciting area of research.
It's what here.
I know. There's so much more to talk about.
I know. I'm so excited for part 3.
Me too. But we have to take a short break for now.
Okay.
Stay tuned.
We're back for the final part of our β-catenin deep dive.
I'm excited to wrap this up. I feel like we've been on quite a journey.
We have. It's amazing how much we've learned.
It really is.
About this one pathway.
Yeah.
I'm really eager to hear about what's happening on the cutting edge of research.
So one area that's really taking off is the development of new tools and techniques for studying the pathway. Remember earlier when we talked about cDNA microarray analysis?
Yeah.
Well, now we have even more powerful technology at our fingertips. Next generation sequencing.
Next generation sequencing.
Yeah.
That sounds pretty high tech.
It is. It's like cDNA microarray analysis on steroids.
Okay.
Instead of just getting a snapshot of gene activity, next generation sequencing allows researchers to read the entire genetic code or transcriptomes in incredible detail.
So they can see not only which genes are active, but also like any tiny mutations.
They can see any tiny mutation.
Or variation.
Or variations that might be present.
Wow.
Yeah, so this level of detail is really revolutionizing our understanding.
It's like a new lens.
It is, on this pathway.
It's like having a superpowered microscope.
Yeah.
That can reveal
that makes sense.
The molecular world in all its complexity.
And with this new understanding, we can start thinking about new therapies.
Absolutely. And you mentioned earlier the possibility of using WNT inhibitors to treat cancer. And that is definitely a hot area of research.
Okay.
One promising class of drugs that's being investigated is called porcupine inhibitors.
Porcupine inhibitors. What?
It's a funny name.
I know.
So porcupine is an enzyme that's crucial for producing and releasing those Wnt ligands.
Gotcha.
That we talked about earlier.
So if you inhibit it, you're essentially blocking...
They're blocking Wnt signaling at its source, at the source.
Yeah, you're shutting down the factory.
That's pretty cool.
It is, yeah.
And that's showing promise.
And in preclinical studies, this approach has shown a lot of promise.
Okay, that's good.
For treating a range of cancers,
such as
including colon cancer, breast cancer, and leukemia.
So there are a bunch of different types of cancers.
There are, yeah. And there are other types of Wnt targeting therapies in the pipeline as well.
Like what?
So researchers are exploring various strategies, such as blocking the interaction between Wnt ligands and their receptors. Or interfering with β-catenin's activity in the nucleus.
So they're kind of like coming at it.
They're coming at it from different angles.
From different angles.
Exactly. Yeah. The complexity of the pathway really requires a diverse set of approaches.
Well, because there's not going to be one.
There's not going to be a one-size-fits-all solution.
Right.
So researchers are really exploring multiple avenues.
It's amazing how far we've come with this.
It is.
It really is.
And we've gone from just like
the very basic mechanisms to actually being able to manipulate it.
And potentially develop therapies.
Yeah,
it could have a real impact.
A real impact on people's lives.
It's really exciting.
So as we're wrapping up this deep dive, I'm wondering what the future holds.
That's a great question.
For Wnt-catenin research.
So one of the big challenges that scientists are still grappling with is understanding how this pathway interacts with other signaling cascades in the cell.
Oh, so it's not...
It's not an isolated system.
It's one pathway by itself.
It's part of a complex web of interactions.
So they're trying to understand how it all...
Exactly. They're trying to understand how it fits into the big picture.
That's cool.
It is. And unraveling these connections is going to be crucial for developing more effective and targeted therapies.
Because you're going to affect more than just that one pathway.
You have to understand the context in which the pathway is operating.
And another challenge must be taking what they see in these cell culture and animal models and bringing it to humans.
So translating what we've learned from those model systems into treatments that work in people.
Because what happens in a dish might not happen.
Exactly. And that's a big challenge in drug development in general.
It's clear that there's still much to learn.
There is, but...
The future of this research is...
The future is bright.
Yeah, it's really exciting.
It's really exciting. The potential is huge.
I think this has been a really incredible deep dive.
I agree.
I feel like I've learned a lot.
Me too.
About β-catenin.
It's been a pleasure.
Thanks for joining me.
Thanks for having me.
And we'll see you next time for another deep dive.